Hello my dear students, myself Meenu Srivastav, and today we are going to discuss about the quantitative measurement of the bacterial growth. In the quantitative measurements, we are discussing about the two type of the count. One is the total count and second is the viable count. And what is the difference is about total count and viable count? Let's see. Total count means that all viable form means living form of the bacteria plus dead cells, or we can say non-living cells or dead cells of the bacteria also included in the total count. Viable means living part, living system, living cells, and the total count living plus non-living cells or dead cells included. And here the quantitative measurements include the three terms. We can measure the quantitatively bacterial count in three terms. First is the cell number measurement, second cell mass measurement, and third cell activity measurement. Cell number measurement, here the cell number measurement, it includes the two types. We can count the cell number into the two form or into the two methods, like first is a direct count, or we can say it's a total count. Second is the viable cell count, or we can say it's an indirect method. Here the direct count or total count, here the direct count includes all the living means viable form plus dead cells also included. That's why it is named as the total count. Okay, so here what is the thing, in the total count or the direct count we are having different methods. So let's see what are the methods are given below. Here the counting chamber technique, in this the counting chamber techniques, the Petrov-Hauser counters is included. You can say that Petrov-Hauser cell counter is same as the hemocytometer. Second method is the Coulter counter, electronic counter method, Coulter counter or electronic counter method. What is the electronic counter or the counting chamber? So you don't worry about it all. We will discuss each and every method in detail. So stay with us. Here the third method is the breed method, and here that is also known as the direct microscopic method. Here in the direct counting method, there are the three methods are included, but in some books you can get another methods also. You can include in your notes and other methods also, but as per my opinion, these three methods are more important. That's why I have discussed here. Now, second form in the measurement of cell number, viable count or indirect method. In the indirect method, there are three techniques or three methods are included. First is the plating technique. In the plating technique, both technique means spread plate technique and the pour plate techniques are included. Here the second method is the membrane filtration method, and the third is the turbidity method. Here now the second in the form of the second measurement of cell mass is here. The measure means the quantitative measure we can concluded or we can measure in the cell mass form also. Here the nitrogen content we can measure if we want to calculate or if we want to quantize the bacterial form, and we can measure the biomass of the bacterial cell. By these two methods, either the nitrogen content calculation or the biomass calculation, we can calculate the cell mass, and with the help of this we can get approx result about the bacterial count. Here the third method, it includes the membrane of cell activity. In the cell activity, we measure the biochemical and the biochemical, or you can say the metabolic activity of any microbial system. By these three methods, we can calculate the bacteria count. Now we will start one by one each and every method. First the counting chamber method, means the Petrov-Hauser cell counter, and second Coulter current method, Breed method and another method also. Now we will discuss the Petrov-Hauser cell counter in the direct method. The Petrov cell counter is similar in the structure as the hemocytometer. As you have done the RBC count experiment, so you can very well know about the hemocytometer, and the Petrov counter is same as the hemocytometer. Here you can see the diagram of the Petrov counter. Here in the Petrov counter there are two chambers. This is not the Petrov counter, this is a box. So I am just taking as a demo. Here the Petrov counter has the two chamber. One is the upper side and second is the lower side. The calculation we have to do on the both side, and we will take on an average value for the direct count. But the thing is that the Petrov counter, the counting, how can we do the counting in the Petrov counter? This is the main task. So let's see what are the structure, what are the chambers, what are the grids are present in the Petrov counter. Then we will see how we can calculate. Just we will start. Here I have already told that in the Petrov counter has the two chamber, upper and the lower, and each chamber has 9 grid. Each chamber has 9 grid. Here I have shown one chamber as a diagram, and here you can see 9 countings, means each chamber has the 9 grid. Okay, here in the grid 5, the grid 5 has 5 into 5, 25 squares. Here the grid 5 has 25 squares. Here 1, 2, 3, 4, 5 and 1, 2, 3, 4, 5 and 5 into 5, 25. So in the grid 5 itself has the 25 squares. Okay, and grid 1379 has 16 small squares. In the grid 1379, 1, 2, 3, 4, 1, 2, 3, 4. 4 into 4, 16. So in the grid 1379, 16 small squares are present. Here the depth of the hemocytometer or Petrov counter, I am repeatedly says the hemocytometer, sorry for that, it's a Petrov counter, and here the depth is the 0.02 mm. Mm represent for millimeter. First of all, we have to calculate the volume so that we can calculate the calculation for the bacterial count. Here we can say the bacterial count. First we have to count all the volume, either chamber, grid, small squares, and that small squares and the other mini squares which are present in the grid 5. Here in the chamber, each chamber carry the 9 grid. Okay, and the one chamber has 1, 2, 3, means 3 mm in width and 3 mm in length, and the depth is 0.02. So the volume of the each and every grid is 3 into 3 into 0.2 is the 0.18 mm cube or 180 nanoliter. In each grid, here I have done the calculation for grid. Each and every grid is the length is 1 mm and its width is 1, and here 1 into 1 and depth is the 0.02. So the total volume is a 0.02 millimeter cube. Now the square in the fifth grid. Here in the fifth grid I am showing that each fifth grid has the 25 square, means in the fifth grid 1, 2, 3, 4, 5 and 5, 1, 2, 3, 4, 5. 5 into 5 means 25. Means the fifth grid has the 25 small squares, and these 25 small squares, each square has the length is 0.2 mm and that 0.2 mm length and width as well, and here the depth is 0.02. so the total volume here the 0.0016 mm cube. This is the main important thing which you have to keep in your mind. Now this the square 1379, each 1379, that is the length each small square is 0.25 and length, width and the depth remains same 0.02. Here that volume is 0.00125 millimeter cube or 1.25 nanoliter. Here the small square, 16 small squares, means in the 5th grade each 5 squares, each 25 square, the 25 squares is present in 5th grade and in the each square is having 16 small square. I have written in the pens with the pencil 1, 2, 3, 4, 1, 2, 3, 4. So there's a 1 square carry the 16 mini square. Okay, so the length and width is the 0.05 and the 0.05 and the depth remains same 0.02. So this is the volume 0.00005 millimeter cube and 0.05 nanoliter. Here, first of all things, the main is dilution. The dilution which we have to keep in your mind that we have to take the dilution either 1:10 or the 1:20. Now, what is the 1:10 and 1:20? The students are generally just getting confused about this 1:10, 1:20. So, I will show you how is the 1:10 and 1:20. Here, when you will take the 50 ml broth in the 450 when you will take the 50 microliter broth in the 450 microliter distilled water, the unit will be same. Okay. And generally the bacterial count we will take as the micron liter. Okay. So when you will take the 50 microliter and the total volume when 450 plus 50, here 50 is to 500 and this is the 1:10. So the dilution will be the 1:10. If you are taking the 1:20, then if you are taking the 50 microliter broth in 950 microliter distilled water, here dW represent for the distilled water that the 50 is to 950 plus 50, here the 50 is to 1000. Now that is 1:20. Okay, so this is the dilution is 1:20. So for bacterial count in the Petrov counter, you will take either this is the 1:20 or is the 1:10 dilution. I think this is the clear and crystalline clear concept, I think, which I will make you understand. Okay, now the formula for calculation here is the bacterial count. The bacterial count is the total count upon the number of square counted into 1 upon square volume into dilution. First, that is what we have to keep in your mind, that all the grid 1, 3, 7, 9, 2, 4, 6 are not at all useful. We have to consider the 5th grid, and in the 5th grid we have to take for the calculation or for the counting of the bacterial cell in the 3, 4, 5, 1, 2, 3, 4 and 5. Okay. So, in the 5th grid, we have to take 5 squares. Okay. 4 squares in the corner and 1 square in the middle. 4 square in the corner and 1 square in the middle. One thing you have to keep in your mind, that while you are counting the bacterial cell, while you are counting the bacterial cell, so bacterial cell which are the bacterial cell which are the bacterial cells which are in between this chamber boundary, that you have to count all. But the bacterial cell which are outline on the outline or the boundaries on the square, then what should we do? For this, the lower and the right cells, the lower side count and the right count you will not to do. Okay, the lower and the right count you will not to do. So upper only you will count upper and left side. Okay, upper and the left side, upper and the left side, that lower side and the right side you will not consider. Now I will give the example so that you can easily calculate the bacterial chamber. Here we have to calculate both chamber. Here we have to calculate, or we have to take on an average for the for both the chambers. Okay, means for 5th grade of upper chamber and the 5th grade of the upper chamber, we have to take the count, and then we will take on an average value. Okay, so now I will give you one example so that you can easily calculate how you can do the cell count with the Petrov counter. Here I have given one example. The example is the bacterial cells were counted in 2 chambers of the hemocytometer. The count in the 5 square of first chamber was 300. Means when we calculated in the first chamber, of the 5th grade of the first chamber, all the 5 corners we have to calculate, and the total count is 300, while in the second chamber was the 256 and the dilution is 1:20. Calculate the bacterial count. So here first the volume, which I have written here in the volume table, that each square carry that 1.6 nanoliter or 0.0016 microliter. Okay, here the formula which I have given, that the total bacterial count upon that 5 chamber, 5 chamber upper side and 5 chamber lower side into 1 upon 1 that volume. Okay, here the volume is 0.0016 microliter and 1.6 nanoliter. So I have written here the 1.6, and dilution. So dilution they have given is the 1:20. So I have written here is the 20. Okay, so the total count is the 556 upon 10 into 1.6 into 20. So those who are not able to calculate, for those I am calculating this is the 0, that here 10 is 10. Okay, and the 556 is divided is multiplied by 20 and divided by 16, and here you can receive that 695 per nanoliter. Okay, 6 means the per nanoliter bacterial count carry the 690 per nanoliter microbial broth carry the 695 bacterial count. Here the if we want to convert it in the ml or the milliliter, so you have to multiply, and because 1 ml is equals to 10 to the power 6 nanoliter. So when you calculate the 695 into 1 upon 10 to the power 6, so 695 into 10 to the power 6 ml. Okay, so in the 1 ml, that 695 into 10 to the power 6 micron bacterial growth will be present. Okay, so this is the Petrov cell counter. I hope you have completed your doubts, and if you have doubt, kindly ask him in the comment section. Now we will discuss about the Coulter counter method, or we can say the electronic counter method. It's a automated method and it's totally depend upon the changes resistance. Now we will see how it can work. First, it based on the principle that change in the resistance as the bacterial culture in the electrolyte solution. In the complete sentence is in the electrolyte solution. Okay, so here the working model is this. There are 2 electrodes are present between the orifice, beside the orifice. You can say orifice or you can say the aperture. Okay, you can say the orifice or you can say the aperture also. Okay, both words are appropriated. Here the 2 electrodes are present beside the orifice. 2 electrodes are present beside the orifice, and here the electrolyte 1 is inside the tube and the second electrode outside the tube, and both electrodes present in a beaker which have low concentration of electrolyte solution. Here the beaker contains the low concentration of electrolyte solution. The aperture, here the first electrodes present in a small beaker or the test tube it contain or it carry a aperture or orifice. In this, the bacterial suspension is pore and the orifice size or the diameter is too small that it allow to pass out only one cell at a time. Here, when the cells comes out or release out, so due to this releasing the bacterial cell, as the battery flows the electrons, or we can say the battery flows the electricity. When the one cell comes out in the electrolyte solution, there will some change in the resistance occur. So the change in the resistance can measure in the form of automated counter. Here this complete system is attached with the automated counter, and this automated counter count the bacterial cell in according to the change in the resistance, because the battery will give the electricity, but due to the presence in the electrolyte solution of the cells, the resistance will change, and that change will measure in the form of cell number. Okay, so here the cell number will measure in the change of in the term of change in resistance. Here the aperture is too small and to allow only one cell to pass out at a time. When cell pass out from the orifice, the change in resistance between two electrodes are measured. Okay, so change in resistance are counted in the form of cell number. So here the electronic counter method, it's a too fast method, and within a few seconds it can count many of the cells. Okay, so this is the electronic method, and it's a small, we can say it's an automated device, and it can very rapid method also. Now we will discuss the breed method. Now we will discuss about the breed method. This is the last method of the direct method. Okay, so here in the breed method, what we are going to we will do in this breed method. Now let's see. In this breed method, take one glass slide. Okay, so in this breed method, take one glass slide, and in this glass slide, this is clearly wipe out. So no bacteria, no other cells will remain on this. So after this, 0.01 ml bacterial suspension, here 0.01 ml bacterial suspension is spread uniformly in the area of 1 square centimeter. So for this, you will take one glass slide which is wipe out nicely, and if the area will already marked is very good, otherwise you will mark the 1 square centimeter, and 0.01 ml suspension you will spread out. So measured amount suspension you will spread very gently or very nicely. Okay, so the suspension volume, you know about that. This is a 0.01 ml suspension spread in 1 square centimeter. So after this, the smear is heat fixed. In this heat fix, as we have discussed in the staining process, the heat fixed means the glass slide will just rapidly we will pass through the that flame. Okay, not stay with that on the flame. We just rapidly wipe out, just remove or just interact with the flame. Okay, now the smear, this is a process known as the heat fix. The smear is doing heat fixed. Okay, now it is stained. The staining depend upon the bacterial culture, either it's the gram positive or gram negative. According this, we have to stain and examine under the oil immersion lens. Okay, here the oil immersion lens, what is this? In the oil immersion lens, the immersion lens has the radius is 0.08 millimeter. Okay, So here you can assume this is the oil immersion lens, okay, and the diameter of the lens, radius of the lens is 0.08 millimeter, and the area of a circular body is the pi r square, okay. I think you know very well about the area pi r square. So here to calculate the area, the pi value is the 3.14, or you can have the 22/7 also. Instead of 3.14, you can put it here 22/7 also. And the radius is 0.08 whole square. Now the calculation is complete. The answer is 0.02 square millimeter. Now it means, what it means? It means when you will observe the slide from the oil immersion lens, you can observe 0.02 square millimeter area at a time. Okay. Now the area of a smear, what smear area you have drawn? This is the 1 square centimeter, and 1 square centimeter equals to the 100 square millimeter, and this 100 square millimeter means the smear you have spread in the 100 square millimeter, means you and at a time you are only observe 0.02 square millimeter. Then how much time you have to observe the slide? It's a 5000 time. How you get it this? 100 upon 0.02. So you will get the 5000. Means 5000 times you have to calculate or observe the slide, then only you can count the bacteria. But practically it is not possible. So there is another, or there is a easy method. We can calculate the average. So here the number of cells per square centimeter, which the per square centimeter is the smear area which have already formed in the glass slide. In this 1 square centimeter, the bacterial suspension is 0.01 ml microbial cell suspension. Here, when we observe the smear, suppose that if we are observing one area and then we calculate the 20 microbes or 20 bacterial cell at particular area. After that, we will shift the lens and in another area we will count. Suppose that 10 cells we got, and then suppose that 10 cells we got, and another time, third time, we will shift the lens and calculate the cells. Here you will get the 30 cells. Suppose that. Now you will take the average. The average is 30, 20, 50 and then 60. It's a 20. Here the 20 cells on an average means per area, okay, per 0.02 square millimeter area, you will find the 20 cells. But you have to see the 5000 time. So you will multiply this 20 into 5000 and the cell number is 1 lakh cells. Okay. So here here we have discussed that how the calculation is done in the breed method. The calculation is important in the breed method because otherwise the smear fixing and the bacterial taking the bacterial suspension is not a big deal. Okay, the method is the how to calculate the things. In the breed method we will discuss we repeat again that we take one slide and just mark the 1 square centimeter area and 0.01 ml suspension is spread out. Now as we have discussed that 5000 times we have to observe this sphere. So we will take the average and multiply by 5000. So we will get the total number, or we can say we get the approx number in the bacterial suspension 0.01 ml. Okay, so now the breed method is the least expensive or easy method, or it's a rapid method also. We can say it's a little bit laborious process, but anyway it's an inexpensive and the cost effective method. I hope you have enjoyed my lecture. Here In this lecture, we have discussed about the direct method. In the part 2 session, we will discuss about the indirect method. So stay with us and please like, share and subscribe. Thank you.